Hello and welcome to a special episode of Launchpad. This is the It's Not Rocket Science strand. And in this strand we shine the spotlight, or in this case the microscope, onto our researchers in residence. I'm your host Paul Stimpson, and I'll be joined today by Libby Duignan from the University of Liverpool. Libby is a researcher in residence from our second tranche and is looking at advancing the manufacturing of phage therapy in the UK. Now, normally I throw to a quick introduction about the network, but you all know what that means by now. Let's skip that bit and get straight into Libby's project and the fascinating background of phage therapy.
Yes, thank you very much, Libby Duignan, for coming onto the podcast. Could you perhaps introduce yourself?
Yeah, I'm a postdoc at the University of Liverpool. My supervisor is Joe Fothergill, and I work in the field of phage biology and phage therapeutics. I can explain what phages are for people that don't know what a phage is.
Yeah, that would be my next question. So that segues nicely into the next bit.
Well, a phage it's known as a bacteriophage, and shortened to phage, is a virus that infects a bacteria. So it can. They're natural. They're found everywhere. A lot of people isolate them from sewage and wastewater. They attach themselves to the cell surface of a bacteria, inject their DNA into the cell. They multiply within the cell and lyse the cell, so killing the bacteria. So in that way, they can be used as an alternative to antibiotics. So that's why they're used as therapeutic.
So it's been around for a while, hasn't it? But it hasn't quite been as popular as, let's say, like penicillin or something like that. Could you maybe explain why, if that's possible?
So I think that it was been around since prior antibiotics, so the sort of 1920s. It was first discovered, and it was actually like cholera phages that were first discovered, and this was sort of by chance, a bit like penicillin. They saw these halos on plates and thought, oh, could this be used as a therapeutic? And obviously this was just sort of very kind of messy science. Nothing was characterized back then or anything like that. So, but because phages is a natural product, there's like always an arms race against phages and bacteria. So the bacteria evolve against the phages, and the phage evolve against the bacteria because they have DNA as well. And when sort of penicillin and antibiotics came around, they were much easier to process and make in bulk than phages. So phages kind of were sort of lost at the wayside in the Western world anyway. In Eastern Europe, though, they carried on doing sort of production of phages, working on isolating different phages as well in the background. So that's why they're kind of ahead of us at the moment, because they kept going with that science.
Cool, yeah. So I guess another question I was going to ask was, why did we kind of not look at phages? Was it a resource thing, or was it just generally, we found this penicillin works? I guess we didn't anticipate the resistance that will come later on.
Yeah, no one expected the resistance that has occurred now, especially against all those different types, because it wasn't the case that, they were kept in reserve until the last sort of 10 years, when they go, actually, we probably should be keeping these sort of newer ones that it's not resistant against yet in reserve. And so now we're coming to the point where people have infections that can't be treated at all by antibiotics, and that's why sort of we're now looking at what we've got on the back burner of phage therapy. So yeah, we might have a look at that instead.
Yeah, it's funny because my wife, she always refuses antibiotics, and I always ask her why, and she always says, because we're building up a resistance and people don't take them properly, people don't finish the course, and it's having a bad effect. And I'm like, are you sure? But now I've been reading about it, it's actually quite serious, isn't it? So how serious would you say the kind of issue is of resistance at this point?
Sort of one of the stats that's always has been said in the last 5 years is by 2050 we're looking at about 20 million deaths a year based on antibiotic resistance. So yeah, I think it's kind of a silent but deadly, because it's going, these people are dying of infectious diseases. You don't think it's due to the resistance that we've caused. It's just going, they're dying of, I don't know, kidney failure or something like that, and this is down to them potentially having UTI and getting sepsis, and it all adds up in the end.
That's a pretty serious number. Yeah. So I guess from what I've read about you, I've read your profile on Liverpool University's website. You seem quite passionate about this subject. What kind of attracted you to this particular little corner of healthcare research?
Yeah, it's quite interesting, really. Like, I did biomed at uni, and I was drawn to microbiology just because I thought it was so interesting. Depending on the different bacteria that you've got a different disease, different features, and I was firstly sort of drawn to like diagnostics and potentially going into, well, I did go into the NHS for a little bit and work on the diagnostic route. However, I did a master's, and that was at Liverpool with Joe as my supervisor then. And project book, and a few of the projects mentioned phages, and all through my undergrad I'd never heard of phages at all, and I thought, wow, this is amazing, because like my projects during my undergrad, I worked on antibiotic resistance in like microbiology sensors, and thought, oh wow, this is so cool that this thing that is like natural can be used as a therapeutic. And I ended up I had 3 projects during my MRes, and 2 of them were based on phages, one on phage therapy, and then one looking at prophages, which is sort of a second life cycle that you can sort of take with phages, where they integrate themselves into the, like, lay dormant in the genome. So I did a project on that as well, which I found really interesting, and that's what I ended up doing my PhD on. And yeah, so it's really cool how you can have a therapeutic arm to it, but then you can also have these prophages that can lead to like give beneficial traits to the bacteria as well. So it's all like sort of pros and cons.
Okay. I bet that was a bit of a light bulb moment when you're looking at resistance, and then you're like, oh my God, there's something here that can probably answer the question that we have on it. A bit of a wow moment. Yeah,
exactly. I think that I hadn't really thought about it before. I thought, oh, okay, so the idea is to sort of limit the antibiotic sustainability, and limit the antibiotics, but not what could we find that's different. And kind of led into this.
So is there, I mean, slightly just to go back a little bit, is there a scenario where bacteria becomes resistant to phages? Obviously, exposing my lack of knowledge of phages there, I don't really know, but is that a potential scenario?
Yeah, that happens like all the time. But the thing is, because phages are well, it's still debatable whether they're living or not, but they have got DNA, so they're able to mutate as well. So it's the constant back and forward. So if someone was given a phage therapy dose, you have the phages within your body, and they can change to then fight a mutation of the bacteria in a way.
Okay. Yeah.
But also treating someone with a cocktail of phages, so I say 5 phages instead of just 1 phage, and they're all slightly different and affect the bacteria slightly differently, which lowers the resistance potential of the bacteria. And sort of looking forwards into sort of the manufacturing side of things, what's been sort of suggested to the government going forwards is, when you have a cocktail, these cocktails are changed a little bit like the flu vaccine, like changed and swapping phages in and out depending on the strains that are going around at the time.
Oh, okay. That's really interesting. So I guess that leads us to your research and residence project with CPI. You mentioned manufacturing. Is that kind of part of your project with CPI, to kind of make sure it is manufacturable, if that is a word?
Yeah. So that's it in essence, really. It's at the moment, phages are not manufactured at the standards that they need to be for clinical trials in the UK. So they need to be made to GMP standards, which is good manufacturing practice. So, and the more you look into it, the more stringent it gets, because I've been working in the lab in academia for so long, just sort of making my phages as I go along. And GMP is very stringent, and it's all drug development. So it looks at like defining the quality of the product, and also the production of it, and also like you need to define clearly validating and reviewing the documentation during the process, which guarantees that the product is as safe and as pure as possible at the end. But at the moment, it's not done in the UK at all, which means that we can't move forward with clinical trials. We can get phages from outside the UK, but as part of sort of resilience within making drugs within the UK, we'd want to have that based in the UK.
So you've mentioned you've popped into CPI and shadowed. How's that been so far? Just, I guess, being in the labs and checking that out.
Yeah, really interesting actually, because I've only ever worked in academia before, and this is like completely different. When I first had my meeting with them, they were like, "What are you going to be doing in upstream?" And I was like, "What is upstream? what does this mean? And like, where does upstream stop and downstream begin?" And then they have analytics as well. That's all separate. Whereas you just do your one experiment when you're in academia. So that's been really interesting to sort of get my head around. And now I feel like I know where I'm standing, and I know sort of what equipment I'm going to be using, and sort of. Yeah, I've got sort of a team leader in each part as well, so I can sort of go to them and ask for help, which has been really good. And they're just so knowledgeable about. They just get so many different projects in, and they're just very flexible on. Yeah, I just know what they're doing. It's really been really useful to sort of even chat to them about, okay, your project you've got at the moment, and I find out about, Project X from their clients, and this is what they're doing, and this is going to be the end product, and it's going to go into GMP. And I go, oh, what, talking about their current project helps me think about mine and how we're going to be moving from what we're sort of planning at the moment into GMP in the future.
So I guess my final question on this project is, what's your kind of post-project finish goal? Is it to kind of take this to the Prime Minister and say and lobby, I've got the phages, let's go, let's stop those experiments?
Yeah, so the Leicester Phage Research Centre, which is sort of headed by Martha Clokey at University of Leicester, she's kind of doing that at the moment, as well as my supervisor, Joe Fothergill. They've been going to the House of Lords, and not so much lobbying, but like seeing how it works and making it known from government that this is being worked on, and that has led to funding. The Leicester Phage Centre has had a lot of funding recently into sort of manufacturing as well. They're looking at more veterinary-based phages, but it's kind of like all going together. So phage therapy in medical, but also in veterinary and in environmental. It's just the use of phages and letting the government know that it's happening. But me personally, I would really like to sort of, if possible, at the end of sort of this project, I come to the end of my my postdoc at Liverpool as well, and I've moved up to the northeast where CPI is based, which was the plan anyway. My boyfriend's from up here as well, so that's always the plan. So yeah, I've been chatting to CPI and asking about like potential jobs there, because this is their first sort of phage project, and they're looking at sort of working with companies being able to produce phages in the future. So hopefully I'll have the knowledge by the end of this to be able to help them potentially join their company.
It's a no-brainer, really, isn't it? Because it's what the country needs in terms of fighting the resistance that that's probably a bit of an oxymoron, fighting the issue that we've already caused. But we've been looking at our EDI data in terms of who's applied and who's been successful. And in our first tranche that we did, we were quite low in terms of the amount of women that were that applied. We had, I think it was lower than 30%. The second tranche is up to about 40%. But clearly there's like an industry-wide, in academia and in industry as well, there's a bit of there isn't enough women, basically. We're not getting enough women into the. I just wanted to know how you think we could make that better. I don't expect anyone to have the answers to this. It's a bit of an on-the-spot question.
Women-wise, I think that after PhD level, people seem to drop out of academia. From having friends and being in academia myself, I'm the only one out of everybody I did PhDs with that's still in academia. Yeah. And I think it's more an academia problem than a research and residence problem. I think as well, like it's the connection between head down looking at your project in research and then looking at how could it be expelled into sort of working with catapults to make it into you're looking for help in other places, I suppose, because sometimes you're just sort of head down, I'm going to do the experiments that need to be done for this project, without going, how could a company help me, rather than how could another department help me. And I think it's not even knowing about sort of these catapults as well within universities, and also having sort of, now you've got people that have done these projects, having these as like examples, and having example catapults to sort of input. These are the kind of projects that we'd work with, or we're interested in working with as well. Yeah,
I do think it's with this EDI approach. The more diverse you want to be, you have to show that you're diverse in the first place, I think. So, we do have a diverse portfolio of researchers, and if we can, it's the vision of it, isn't it? If you see people that are from your background, then you'll be likely to think, actually, yeah, this could be for me. It's not just middle-aged white people just trying to get away from.
Yeah.
So.
Also, I will just add that I was part of this a pilot scheme that was run by the University of Liverpool and Manchester and Lancaster, I think, but they've now made it sort of wider, open access to all universities, called the Prosper Programme, which is for postdocs to sort of see where their careers can go after academia. And they do sort of people come in and do talks online mainly, and I was just thinking that it's not necessarily aimed for people to go outside academia, but I mean, I think that if you can grow your project and work with external people, that sort of just grows your skill set as well, even working with catapults as well. So I think maybe if you could get involved with that could be good for it as well.
Yeah, definitely. Always looking to do things like that. Libby, I'm going to say thank you very much for joining us. Do you have anything to add, actually, after?
Not really, other than this opportunity has been sort of the best thing that's ever happened for me.
Oh wow!
I always wanted to sort of move into sort of manufacturing of phages, because it's my whole background have been on research, and if it can't be made into a product, it's a bit pointless. And so I think this has been amazingly useful for me to see what happens in industry, and yeah, it's been very good, basically.
That's really good to hear. That's really good to hear. Like we said off tranche one, some haven't got going yet, but some have nearly finished, and they've said similar things, like that the impact of just having this award and going into industry, going into the labs, it really helps. I think a couple of people have had promotions and stuff since. So yeah, all the best. I think we'll definitely catch up. Maybe in a few months, 6 months to a year, see how you're getting on with your project. Hopefully you're queen phage by then. But yeah, thank you very much for coming on.
How good was that? Thanks so much to Libby for sparing the time to discuss her project with me today. Her project with CPI, part of the high value manufacturing catapult, of course. It's brilliant to hear how much of an impact our researcher in residence scheme is having, and generally it's just great to hear someone discuss a topic so passionately. We'll catch up with Libby later on in the year to see how she's getting on with her project. One thing to mention before I go, I mentioned it last podcast, the launch of the abstract, the first issue, is out now. We've done it. You can read online via the link in the description of this podcast, or head to our socials, which is just Innovation Launchpad Network plus on LinkedIn and Twitter, X, and look for the pinned post. There are physical copies of this available, so drop me a line if you do want one. My email will also be in the description. The first issue of the abstract is all about the first tranche that we did of our researchers in residence scheme. So there's plenty of features in there from our researchers themselves, our investigators. It's a cracking little read. So do pick one up virtually, I suppose, or email me if you want to pick one up physically. That's all for this month, and we'll be back again next month with another podcast. Bye-bye.